And so next, I would like to introduce Luis Alberto Vega. He got his Ph.D. from WashU in the lab of Dr. Michael Caperon, studying Streptococcus pyogenes. He did some postdoctoral work in the McIver lab at the University of Maryland, and he is now a postdoc in the Flores lab at UT Health. And today he's going to tell us about an integrative conjugative element that contributes to skin and soft tissue pathogenicity of emergent antimicrobial-resistant group A strep. Take it away, Louis.
Thank you, Patricia, for the introduction. And yes, like Patricia said, today I'm going to share with you all a story about how an integrative conjugative element of group A strep contributes to virulence of a antimicrobial-resistant strain of group A. So jumping right into the topic, mobile genetic elements are the segments of DNA that mediate horizontal gene transfer among bacteria. And these include phage, plasmids, and integrative conjugative elements. And as elements that horizontally transfer among bacteria, these are the primary vehicle by which antimicrobial resistance genes are maintained and disseminated across bacterial strains and bacterial species. So it's very important to study them, as Shreya pointed out, given the impending crisis with rising rates of antimicrobial resistance and the threat that this poses to public health. Integrative conjugative elements are of particular interest precisely because of several of their traits. One is that their large size, which ranges from 10 up to 80 kb or more in size, kilobases or more in size, which allows them to encode a great variety of gene content that is mosaic in nature, meaning it can incorporate the gene content of chromosomally encoded content of the bacteria that they are integrated in, as well as genomic content from other mobile genetic elements such as plasmid and phage. And being chromosomally integrated yet mobile allows ICE to both vertically transfer and horizontally transfer among bacteria, which means that they have great potential to both contribute to antimicrobial resistance and bacterial disease potential. And they do that through a variety of ways. They can do it by both encoding virulence and antimicrobial resistance genes. So you could have a situation where an ICE integrated into one bacterial genome can incorporate through recombination a virulence gene such as a toxin, and then transfer that into another bacterium when it excises, conjugates, and integrates into that genome. They can also encode virulence genes that influence antimicrobial resistance directly, such as the case of siderophores or membrane transporters that can not only have an effect on virulence or that are required for virulence in certain bacteria, but also allow the bacteria to efflux antibiotics. And ICE can also indirectly influence virulence just by virtue of their integration or other unrelated gene content, either altering the differential gene expression of core chromosomal genes or contributing metabolic gene content that alters fitness, as well as providing the bacteria with the ability to occupy novel niches, either through gene content that now allows them to survive in those niches, or by virtue of the antimicrobial resistance itself, which allows them to survive and outcompete susceptible bacteria. In one bacterium in which mobile genetic elements featured prominently is group A strep. So group A Streptococcus, or GAS, as I'll refer to it, is a specifically human pathogen. It's unique. It only infects humans, and will, for the most part, asymptomatically colonize the nasopharyngeal tissue of its host. However, it can also cause infection, the most common being the nasopharyngeal, symptomatic infection, the most common being the nasopharyngeal infection that most of us are familiar with and commonly referred to as strep throat. But group A strep can also cause invasive and even life-threatening infections in the blood and subcutaneous tissue and muscle. And the way it does is able to colonize all these tissue sites is by the expression of a multitude of virulence factors. And these are differentially expressed across gas serotypes. And also different gas serotypes have different combinations, alleles of these virulence factors. So both this differential gene expression and genoplasticity of group A strep, which includes the incorporation of mobile genetic elements, is what contributes to its versatility as a human pathogen. Now, more specifically, with regards to mobile genetic elements and what we know about how these influence virulence, we know that the ones that are the best, characterized and most well studied are prophage or phage that have contributed a variety of exotoxins, DNases, and other virulence factors to group A strep. And here I'm sharing with you this figure from a publication where the authors indicated all the integration sites on the Streptococcus on the group A genome into which various phage have integrated. They also indicate on the outside of the ring the various toxins and virulence factors that these phage have contributed, as well as on the inside all the different serotypes in which they are found. So clearly, phage have contributed a large amount of virulence-associated gene content to group A strep. We also know that presence of these phage can affect differential gene expression, particularly of core chromosomal genes involved in metabolism or virulence. And there's also research to show that ICE are associated with disease potential in group A strep. The best case of this is the region of difference 2, or RD2, that was identified in M28 serotype strains that are primarily associated with puerperal sepsis cases. And what the researchers who have characterized RD2 showed was that if they conjugatively transferred RD2 into other serotypes, these strains now had an enhanced ability to colonize a mouse model of vaginal infection, showing that clearly ICE is contributing some sort of virulent phenotype to gas. So this gets us to my part of the story, which is looks at the association of antimicrobial resistance with ICE and virulence. So antimicrobial resistance in group A strep has not been particularly well studied, simply because, or primarily because, group A strep are universally susceptible to beta-lactam. And so there's not been that much focus on antimicrobial resistance in strep. However, epidemiological data shows, and this is a screen grab from the webpage of the CDC's active bacterial core surveillance, shows this epidemiological data shows that over the past decade, there's been a substantial increase in the number of invasive infections due to group A strep. And a lot of those infections are associated with increasing rates of macrolide, aminoglycoside, and tetracycline resistance. And these are all second-line antimicrobials that can also be used to treat group A strep infection in those people that are allergic to beta-lactam. So that raised level of concern in terms of the problem that this could engender for public health. And a majority of the contribution of this antimicrobial resistance in group A strep is due to a subset of serotypes. This is a graph from a publication using that same data collected by the ABCs from the CDC to show the increase. This graph increase shows the increase over time of macrolide resistance among these serotypes, which are the ones that are primarily responsible for resistance among invasive isolates. Research done by our own group showed similar strong correlation between invasiveness and antimicrobial resistance. So what this figure from our publication shows is that on the y-axis, you have an invasive index, which is simply just a measure of invasiveness, consisting of the proportion of invasive isolates versus non-invasive isolates of each distinct serotype that we examined. And then on the x-axis, you have the frequency of antimicrobial resistance identified in those isolates. And what we see is that just like the CDC data, we have a subset of serotypes that are highly invasive and highly resistant. So those include M75, M11, M77 isolates, and M92 isolates, which don't even factor in the figure because they are almost exclusively isolated from invasive infections and have very high rates of antimicrobial resistance. And that also follows along with the CDC data, which shows that M92s are actually the serotypes that have shown the greatest increase in antimicrobial resistance in invasive infections over the past decade. So when we look more in depth at the source of the antimicrobial resistance in these serotypes that are highly invasive through using whole genome sequencing, we found that the antimicrobial resistance in these strains is encoded in a diversity of ice. So this figure just illustrates the different ice in each one of these serotypes in which the antimicrobial resistance is encoded. And I just want to draw your attention to M92s, which contain a ice of recent emergence, ice by M92. We hypothesize this is an ice of recent emergence because all of the antimicrobial resistant invasive isolates that we have identified thus far encode a highly conserved copy of this ice, which is what suggests that it's a recently emerged feature of the M92 serotype genome. So this raises the question for us whether this ice is responsible or contributes significantly to the virulence of this gas serotype. So our hypothesis then was that the gene content encoded of ice by M92 is contributing to gas virulence by influencing global gene expression in gas. And to do that, to test this hypothesis, first we wanted to compare contemporary M92 isolates to a historical isolate that is susceptible and of a similar genomic background. And for that, we used the MGAS270 strain, which is also an M92 serotype strain, but that contains does not contain ice by M92, nor does it contain any other antimicrobial resistance. We also tested our hypothesis by generating isogenic mutant strains in the ice in both of these backgrounds. So to that end, we used a counter-selectable 11-sucrase cassette to induce excision and loss of the ice in an M92 contemporary isolate, and then introduced the ice by M92 into the MGAS270 background by conjugative mating. And we were able to achieve integration of the ice at the same gene locus in the MGAS270 background as it is found in the contemporary M92 background. So first thing we did was to look at the invasive phenotype of these strains using a subcutaneous infection model in mice. And what we see is when we compare the ice-negative MGAS270 strain to the positive M92 strain, we see a distinct difference in the formation of necrotic lesions at the site of infection. And if we monitor this over the course of 7 days, you see a statistically significant difference in the size of the necrotic lesion, suggesting greater virulence and invasiveness on the part of the ice-positive contemporary M92 strain. We proceeded to make the same comparison now using our isogenic ice-mutant pairs. So you have the contemporary M92 and its delta ice knockout versus MGAS270 and its ice-positive mutant. And what we see is a statistically significant difference in the necrotic lesion size formed in these mice. And we see a similar trend, though not significant, in the lesion size comparing the M92 strain to its delta I knockout. When we looked at the colonization burden, and these experiments were done with a lower infectious dose to the previous experiment, so that we could also not only look at virulence, but also the ability to colonize, if there were any differences in the ability to colonize the skin tissue between these strains, we see, first of all, that the contemporary M92 colonize or infect the skin at a significantly higher infectious burden than do the historical MGAS270 background strains with or without the ice. When we look at individual comparisons, so using comparisons in individual mice, since we contralaterally infected them with an ICE positive and ICE negative strain, isogenic mutant strains, we see that presence of the ICE significantly affects the tissue burden in both the M92 and the MGAS270 background. So clearly the presence of the ICE is contributing to the ability to colonize and cause invasive disease in the skin of a host. So next we wanted to test our hypothesis that ICE by M92 influences global gene expression in the M92 background. And to do that, we did RNA-seq analysis on in vitro cultures of our isogenic strains. So comparing M92 to its delta ICE mutant, we saw differential expression of 60 genes in an ICE-dependent manner. And in the MGAS270 background, we saw differential expression of 200 genes. And comparing the two data sets, we saw 44 genes that were similarly differentially expressed in both backgrounds in the presence of the ICE. And that's what I'm showing here in this graph. We see a strong correlation in the level of differential expression of these genes, and these include genes involved in metabolism, such as the lactose utilization operon and the arginine deaminase pathway, but also factors involved in colonization in virulence, such as the lactate oxidase identified, encoded in LCTO, as well as a chemokine protease encoded by SPICEX. We also wanted to look at, given the difference in the number of genes differentially expressed in both backgrounds, as well as the phenotypic difference between the contemporary M92 and the historical MGAS270 background, whether there was a contribution from chromosomally encoded gene content to this phenotype and to the differential gene expression. And we did. We actually saw that if we compared, so here on the y-axis, you have the delta I transcriptome relative to the MGAS270 transcriptome in vitro, and on the x-axis, the M92 to the compared relative to MGAS270 transcriptome. We see a strong correlation in the differential expression of several genes, which include multiple virulence and factors that include adhesins, such as FPPA and SCPA, virulence factors like M protein, and cytotoxins, such as NGA and SLO. So that means that also there are different core chromosomal differences in these emergent M92 strains that are highly invasive and resistant, that are potentially contributing to the phenotypes that we see. So finally, we wanted to validate whether this differential gene expression we see in vitro occurs in vivo, and for that we used an ex vivo model of infection in which group A strep are exposed to human epithelial keratinocytes in culture. These are primary cells grown in culture. Bacteria grown to exponential phase are allowed to adhere to the keratinocytes. Then the keratinocytes are washed so that only adherent bacteria remain. The bacteria are incubated, co-incubated with the cells for another period of time. then they are harvested and the cells are lysed to isolate the preserved mRNA, which is then measured by QR, in this case, by QR qPCR. And what this assay showed to us was, first of all, was a couple of things. First of all, we see that there is differential gene expression associated with the presence of I-SPY M92, specifically of genes like LTPO, the M protein, and the cytotoxin SLO. But interestingly, this differential gene expression was also dependent on the genomic background. So we only see significant differential expression in the M92 background, not in the MGAT270 background, with the potential exception of SLO, which, however, though it shows a trend for enhanced expression in the presence of I-SPY M92, wasn't statistically significant. Also, when we compare differential gene expression in the M92 background relative to MGAT270, we see that there is also a contribution from the genomic background in that not only are LTPO, M protein, and SLO increase in expression in an ICE-dependent manner, there's a basal expression level that's greater of these transcripts than in the MGAT270 background. Furthermore, the bicep chemokine protease is enhanced in expression in the M92 background, independent of the ICE. So what these data have led us to conclude is that I-SPY M92 gene content influences gas-whole gene expression, and in conjunction with core genomic traits of the contemporary M92 strains, is contributing to the enhanced skin and soft tissue virulence that is observed in M92 isolates. So potentially, it is the activity of both gene content on the ICE and alleles specific to the M92 background that are resulting in differential in a global gene expression program that enhances M92 virulence. So some of the future directions of this project include the following. One is we know there are multiple transcriptional regulators encoded on I-SPY M92, and we want to know, does the activity of these transcriptional regulators influence, is that what's responsible for the differential gene expression that we see in an ICE-dependent manner? And we can look at that by mutagenesis of the transcriptional regulators in the ICE or introducing them into the MGAT270 background all by themselves. We also want to know what contribution can I-SPY M92 make to virulence in a different genomic background. So if we were to put it in other serotypes that have different patterns of disease potential, whether they are skin specialists or nasopharyngeal specialists, will the presence of ICE also alter their pathogenicity? We can also look at whether regulation or expression of the antimicrobial resistance genes plays a role in how I-SPY M92 influences virulence. Is there a regulatory response to the presence of antimicrobials? And finally, we also want to look at these other ICE. So we know that there are different ICE encoding antimicrobial resistance in these strains that are being isolated from invasive cases of gas infection with increasing frequency. So are those ICE also contributing to the virulence of the strain background? And with that, I would just like to acknowledge both the institutions who supported this research, the funding. All of this work was conducted under the support of a P32 training grant. Primarily also would want to thank the people involved, my mentor, NCI, Dr. Anthony Flores, members of the lab, which I've highlighted, who directly contributed to this project. My co-mentors, Dr. Sam Shelburne and Dr. Cesar Arias in the T32 program, and also our close collaborator, Dr. Carol Baker. And with that, I'm happy to take any questions.
Hi, Luis. This is Terry Kohler. Nice talk.
Thank you, Terry.
So I'm wondering, so the ice-related or ice-associated differences in tissue burden.
Yes.
I'm wondering if there's an overall fitness cost to having ice. And so in vitro culture and any condition, do you see a difference in growth rate, for example, between the ice plus and the isogenic ice plus and ice negative strains?
Right, yes, thank you for that question. I did not include that data here for interest of time, but it will be in the manuscript that we're submitting. And no, we don't see any differences in vitro when we do in vitro growth. And all of our in vitro RNA-seq was done using exponential phase cultures. And yeah, we did not see any difference in the rates of growth due to the presence of the ice. So that makes it more of an interesting question because yeah, one would expect traditionally that these mobile genetic elements incur some sort of fitness penalty or growth penalty by virtue of the cell having to replicate them. But I think the fact that they're so well conserved epidemiologically, according to the epidemiological data we have, suggests that that penalty is somehow being compensated for by a contribution to virulence. And we think that's what we're seeing here with our data. But yes, thanks for the question.
Have you tried, for in vitro culture, have you tried different growth conditions? Have you tried like a synthetic minimal medium, or are you just growing in rich medium?
Right. So yeah, we've only done growth in rich medium. That's definitely a question moving forward. Yes, if we were to move into other, we're even interested in repeating potentially that our ex vivo model doing RNA-seq with the ex vivo model, because that would potentially also tell us more. Or if we could do RNA-seq from in vivo samples in the mouse, that would tell us even more as to how this ice is influencing global gene expression directly in the host. Definitely.
Thanks.
This is Magnus Hook. Can I ask you a question?
Sure. Thank you, Dr. Hook.
So we're working in our lab on ice like mobile genetic elements, and the question that I have is really in comparing different ice structures, do you find any putative virulence factor in the actual ice, novel virulence factors, such as cell wall anchor proteins, an extra sortase, or things like that?
Yes, so we've looked at what the gene content that's there. There are some putative cell wall proteins, uncharacterized cell wall proteins. We've also considered that as a possibility of taking some of those operons that are in ice by M92. The biggest chunk of ice by M92 is actually all of the conjugation machinery. That takes up nearly 40% of the ice, which there's also the possibility that that conjugation machinery in and of itself is somehow affecting virulence. We don't know. We've just focused on this project on looking at the differential gene expression question, since that was something that nobody had looked at in terms of resistance encoding ice in group A strep. But definitely that's a possibility that there's other gene content on the ice that we know is just, labeled as putative, membrane proteins that contribute. I know that in RD2, that other ice that has been investigated in gas, which doesn't encode any antimicrobial resistance, they thought that they had a hunch that there was a putative membrane protein that was responsible for the vaginal colonization phenotype that they were studying, and deletion of it didn't have an effect. So they also their work also pointed us in the direction of transcriptional regulation, because when they looked at one such putative membrane protein, they didn't find an effect on virulence. But yeah, no, that's a great question. Thank you, Dr. Hook.
And I don't know if Srishti Arora, who is actually leading these efforts in our lab, is on the call, but you guys may want to get together and compare notes.
Sure, definitely would love to. I appreciate that. Thanks.